So for today's Journal Club, I'm very excited to have Kilian Pohl joining us from Stanford. And Kilian is the founder of the Computational Neuroscience Lab and is an associate professor in psychiatry and behavioral sciences at Stanford. And he also holds a secondary appointment as program director of biomedical computing at SRI International. Kilian received his PhD in computer science and AI at MIT, and before Stanford was at Harvard, UPenn, as well as IBM Research. His interest focuses on developing unbiased deep learning models for improving the mechanistic understanding, diagnosis, and treatment of neuropsychiatric disorders. And today, Kilian will be sharing some of his recent research on training confounder-free deep learning models. So Kilian, welcome to the AIMI Journal Club, and please take it away.
Hi, thank you so much for the nice introduction. I don't have anything to say anymore. Can everybody hear me? Is the sound okay?
Sound is perfect.
And you see my little arrow here when I'm pointing around?
Yeah? Yeah, there we go. Yeah.
Great. Yeah, so I was asked to present a recent publication of ours that was entitled like this talk, but I'm actually going to talk more in general about machine learning, deep learning technologies, and how we can possibly model biases and confounder in the context of neuroimaging studies. But before I do this, I have to thank all the funding resources that basically fund the research that I do, as well as those that work with me. So my view of the world is very much, I'm part of the Department of Psychiatry, and so my view of the world is somewhat biased, so to say, by that department. And so neuroscientific studies, how they generally are done is somebody collects, for example, MRI images of two cohorts, and then I get those images and extract anatomical connectivity and resting state measurements from those images in order to support a certain hypothesis that identified group differences between cohort 1 and cohort 2. Now that's a very conventional approach of performing analysis because it's partly because the findings are relatively, in most cases, easy to explain. To give you an example of that, we just published at JAMA Psychiatry about white matter integrity within the Incanda cohort. That's the study was about 600 subjects at baseline between the ages of 13 and 21. You'll hear more about this study. And we basically tracked their white matter integrity throughout the span of the study as they were aging. And so what you see here in kind of light green is the normal aging trajectory of white matter, whereas in individuals that before they started drinking followed that trajectories, but then once they started heavy drinking, that trajectory deviated. And it deviated more the earlier they started heavy drinking, that's shown here. So those kind of studies, that's, for example, a quick example of how we can use this traditional design to push forward more about increasing the understanding how alcohol drinking, for example, influences brain development. Now, this kind of inference, this kind of analysis is really limited by the various prior assumptions that we're making throughout the study. And so our goal is to further expand that. First, we would like to not only analyze one data set, but multiple data sets to increase sample size, which often improves power, as well as heterogeneity of the data to allow more generalizable results. Furthermore, we would like to go away from that concept of extracting a few measurements that are really tailored towards a specific hypothesis, but extract many measurements that can increase the accuracy in characterizing our cohort. Finally, once we have extracted many measurements, we have the problem that these simpler models that traditionally are used in neuroscientific studies cannot handle that complexity anymore. So then I started to look into machine learning in order to identify or predict which of, for example, MRI images are associated with one of the two cohorts. Now, that kind of inference is more difficult to interpret, so that what we have really focused on is to associate inference with an informative pattern that we validate by looking at how accurate does this pattern predict, for example, disease outcome, and associate significance of that pattern with that. To give you, so when you do this, then the next question is, how do you model bias? And I first will talk about bias because in the machine learning community, that's kind of what people are used to. And then in the next couple of slides after that, talk specifically about confounders and what I mean by that. So the way that in fair learning, machine learning, deep learning, people talk about bias or data bias is that basically the data set that is analyzed is confined to a specific cohort. For example, I want to identify sex differences, but the samples that I'm analyzing are mostly Caucasian. So the question is, how does these results generalize to populations that are not covered by this cohort? And there are a variety of approaches that have been proposed in this domain. One of them is data stratification, data matching, and recently invariant feature learning, just to mention a few. Basically, what this comes down to is you have a skewed data distribution, and you now try to do inference based on the skewed data distribution, which will bias your findings. And so that's kind of, so what all of these technologies essentially do is remove the direct association between your measurements and your bias. Now, in neuroscience, we have a little bit more complex scenario. There's not only a skewed data distribution, but there's also a bias towards my label or the task or the two cohorts that I'm trying to analyze. So there's an intrinsic dependency with the label, and hence that leads to erroneous conclusion. What do I mean by that? Here's an example. For example, here we have a study that consists out of controls and those that are HIV positive, where my control cohort is significantly younger than the HIV cohort. We also know that cohort accelerates essentially aging, so that that there is a interdependency there. So by removing, so now our goal is essentially move the direct association between my measurement and my confounder age, while preserving my indirect association with respect to the HIV specific aging of the brain, if you want to label it as that. So one way of achieving this is by forcing conditional independence with respect to controls by removing essentially the normal aging effect and then viewing what essentially is the residual and associating that with HIV positive. So how do traditional approaches do this? Essentially, they extend this concept that we currently display here, this kind of analysis, by including a regression analysis. So the measurements are fed through a regression analysis that removed the confounding effect from the measurements, and then you perform this kind of machine learning. We did something like that in a scientific report that we published in 2017. So here's again this example where you have an aging effect in green. This is your kind of your healthy population, where in black is, in this synthetic example, is the disease cohort. And you see here that there is an aging effect, but the aging effect is more pronounced in my disease cohort. So if I'm just now performing kind of group separation based on the score that I'm giving, I cannot distinguish the two. So people perform a regression in order to regress out the confounding effect. Let me go back one. So when you do that, you basically fit a general linear model, for example, just to your control cohort. Now the control cohort contains noisy measurements so that there is a family of solution that you can use in order essentially to remove the aging effect. I choose now here, for example, this one with the red line, which depends again on your regularization, which specific solution you're choosing. Then you regress out your confounding effect, and now your classification is much simpler. When you do this, now you can much better separate the two cohorts. But as you see, it's not perfectly separated. And the reason why in this specific synthetic example, it's not perfectly separated is because your regressor was kind of an arbitrary criteria that you choose and not necessarily associated with a analysis, with a postponing analysis. So what we are proposing in this paper is actually to optimize regression and the proceeding analysis jointly, so that your analysis, your proceeding analysis is basically modeling regulates your search space. So in this case, we find a better solution that in this case perfectly separates those two cohorts. And we applied this concept to data from the Encanto study, which contains 671 individuals that minimally drank at baseline and 34 regular drinkers. And the scores that we had were structural measurements as well as diffusion measurements. And the two groups were matched, but with respect to age and socioeconomic status. So when we put this into a Sprouse classifier that was defined by the L-new norm, the row scores just got us an accuracy of 57%, where we see essentially from the sensitivity and specificity, it labeled almost everybody as healthy or controls in this case. And the sequential approach slightly improved the accuracy, but was still fairly low by 62%. When we did the joint modeling, we greatly increased the accuracy to 75%, almost 76%. More importantly, when we now measured the accuracy of this predictor just on a matched data set, then our accuracy by our joint approach stayed stable around 76%, where it dropped for the sequential approach. So what this means that even with this low accuracy, relatively low accuracy, it was still impacted by age and SES as possible confounders. And this is also, we then did a follow-up test, an age test, where we left the labels as they were and just looked at if we use those labels to distinguish younger from older cohorts, can we do that? And in the case of the raw and the sequential approach, we were actually able to classify those two groups with significant accuracy, where this wasn't possible with a joint approach. So that was the only one whose results seem to be not as impacted by age anymore. So kind of the lesson from this slide is, even if you think that you have taken care of the confounder in your analysis, be careful, because residual effects within your data might be picked up by powerful statistical models, such as those proposed in the machine learning community. I promised you in my earlier slides that I'm not only presenting methods, but that I'm not only predicting accuracies, but that I'm also associating those with patterns. And so there were 8 regions associated group differences between regular drinkers and non-drinkers. And the regions were also associated in the alcohol literature with alcohol drinking, to be impacted by alcohol. Interesting, when you just do a volumetric or an ROI specific analysis between those two groups, only 4 of the 8 showed really group differences. So what's important there to highlight is essentially that here we look really at the combination. We're looking at a pattern of regional measurements instead of individual ones. And the individual ones, if you just look at one-to-one correlation between your cohort, distinguishing your cohorts and your ROI measurements, this might be not as informative as looking at really a pattern of ROI measurements. Now, so far what we have kind of focused on is a model, a machine learning model that consisted of where we extracted measurements from data sets, and I showed you a variety of ways, or gave you examples of how removing confounding effects. One problem of this model is that we are still a priori constrained to the measurements that we extract with respect to our analysis. And that's where kind of like the recent development of deep learning, and especially end-to-end model, can really help us to overcome that assumption, where we essentially optimize not only the prediction, but also the type of features we can extract from the data by directly feeding the imaging data into our model. When you do so, when you do that, then the question then becomes again, how do I model confounders? And this kind of analysis, and this analysis was spearheaded by Professor Daily and Dr. Zhao, so they get a lot of the credit of the work that I'm going to present in the preceding slides. So take kind of your vanilla CNN deep learning approach. So you have the image as an input, you extract certain features, and then you do a prediction with respect to that you're trying to predict which class this specific image is assigned to. So how do we now account in this framework with respect to confounders? Because we have not defined a priori our measurements that we would like to analysis, but it's all part of the optimization. So what we did first was a post hoc analysis. We trained our model, we got an encoder, predictor, we got a set of features, and now from those set of features, we could basically fit general linear model to test which of the individual features was significantly correlated with, was significantly confounded or correlated with the confounding values. And those that were, we could essentially mark as 1, or those that weren't, we could mark as 0. Then we can flip it, and once we do partial backpropagation, omit all the features that we that were significantly correlated, and basically just compute our salience map, the importance of regions in the predictions based on features that were not significantly correlated with our confounders. When we did that with respect to the ABCD study, when we started this analysis, the study consisted of 4,000 girls and roughly 4,000 boys. Just now, when they closed out baseline, they had more of like around 12,000 samples. These two, but the girls and boys that we had access to at the time were significantly different with respect to age. Girls were significantly younger than boys, and pubertal development scale where the girls had higher values than the boys. When we applied this model without without omitting the effect of confounders to the data, what we saw here that essentially we had a pattern consisting of temporal lobes, subcortical regions, and cerebellum and corresponding white matter. Now, in the next image, we moved those features that were associated with the confounder, and you see that the pattern is much more focused. Interestingly enough, of the top 10 regions, most important regions, most of them were actually associated. It showed volume differences between the two groups, but within the cerebellum, most regions within the cerebellum, which also were associated with pubertal developmental, showing a pubertal developmental effect. Sorry about that. But this kind of approach is kind of unsatisfying because it's kind of a post hoc analysis. What you want to really do is to extract features while you're doing the training and make sure that those features or that training accounts for confounders. And so the question is, how do you want to do that? And the way that we initially started to solve this problem, and this was also the approach that then was published in Nature Communication, was by adding essentially a component to our architecture an adversarial component that's tried to predict the confounding value from the features itself. And so in this adversarial framework, we tried to find features that were most predictive of our cohort assignment, while at the same time being minimally predictive with respect to the confounding value of each individual's subjects. Now, doing that is so you can't have extract features that predict label Y while they're invariant to the confounder C. That can't be true. But what we showed is if you actually confine here your confounder module with respect to a specific sub cohort of your training data set, so which we call Y conditions, so for example, just the controls, then and perform your analysis up here just with respect to that limited cohort, then you can show that F is going to be mean independent with respect to the confounder C. Now, I'll give you an example here. We applied this to a data set of containing HIV, 120 HIV subjects and 223 controls that were significantly different with respect to age, where the controls were older than the HIV population. Now, if you just apply your 3D CNN, your Confounder model, naive model, you got an accuracy that was around 71%, and the one that we proposed was slightly higher. More importantly is that our precision and recall in our approach was much more balanced versus here your 3D CNN model basically assigned most cases to being controlled. And that's also then shown if we are doing now computing the accuracy score with respect to a data set that's matched with respect to the confounder age. What we saw then is that the accuracy drops for our naive approach, but when we model the confounder during training, we achieve roughly the same accuracy. And again, also precision and recall is preserved, where here even the difference between the two of them significantly more increased. I said earlier on that most of the individuals were of the HIV subject were labeled as controls by the naive approach, and you can see that if we further divide this matched data set into young and old, then here the accuracy is significantly less for the naive approach than ours, because the younger HIV cohort were basically, the approach couldn't distinguish between younger HIV cohort and the controls. And this is also shown here visually in this graph. And here, if you look at the feature space, you can see that the feature space by the naive approach shows a cluster of individuals that are older here and younger down here, where this is not the case for our model that accounts for the confounder. And the impacted region here also contains much more the ventricles, which are associated with aging, where here we have we have a much more focused pattern that is more informative. We also, in the conference paper, we also compared it to various other approaches. We expanded the baseline comparison and also applied it to non-imaging data. And again, we were showing that our approach, the features that were extracted were least correlated at the end with the confounders. And this is also nicely shown here in the plot of the training accuracy, where here the confounding accuracy, the correlation with the confounders nicely goes down, where here the naive approach, stabilizes above, and the multitask approach, which actually achieved the highest AUC, actually increases as the training goes on. It increases its correlation to the confounder. Here's another data set that we tested it on, again, at Canada, trying to distinguish girls and boys, where now pupil development scale is the confounder. And now we have here three possibilities to essentially define on which cohort we would like to model or extract the confounder from. And this would be, I can take all samples, which then gives a fairly low accuracy as expected, slightly higher if we are just confining the confounding analysis just to the girls, and it increases when it is with respect to boys. And that's simply because at the start of the study, the girls already had much higher PDS scores to start out with, so their range of scores was not as well distributed as for the boys. Now, if we are looking on a matched data set with respect to boys and girls, what we can see is that our naive approach, again, it's basically replicating the finding that we previously found on the HIV data set, where the naive approach drops in accuracy, whereas the our CFNet, which models the confounders, has a similar accuracy. And if we are now further refining it to those with lower PDS scores, we have a significant difference between the naive approach, because essentially by the naive approach, girls that had low PDS scores were generally labeled as boys, because again, the approach could not distinguish that, where we could model that in our setup. Non-MRI images, we also looked at those by predicting the bone age from hand X-ray images, where the images were from the RSNA challenge of 12,000 children, where here, now the interesting thing is we want to predict age, which is a continuous and not a discrete output predictor. And our confounders is boys and is sex, basically, as boys were significantly older in this study than girls. So here we define our Y condition subset with respect to a age span between 75 and 175, and we're showing that in this subset we decorrelated with respect to gender via bootstrapping. And so what we can see here is when we did the naive approach, essentially girls were estimated, their bone age were slightly overestimated, and where for boys it was slightly underestimated. And when we applied our confounder-aware deep learning model, but trained the adversarial confounding component on the entire data set, we see there's no difference between the two approaches. However, if we are confining the adversarial component with respect to the Y condition cohort, then we see here that the girls and boys, the prediction pretty much follow. There's no great bias across those two cohorts anymore. And interestingly, also the attention maps that we generated, our approach produces attention maps that much more focus on anatomical regions, versus the naive approach has a much more diffuse map. So this completes kind of the presentation of the article that you were interested in, and I wanted to shortly show you also what we have done since then. A much more, I find, elegant solution was spearheaded by Asana Daily and Mandy Liu. Mandy is a graduate student in the computer science department that wanted to integrate in whatever architecture you have, essentially integrate another layer between two layers that would model the issue with confounders. So called metadata normalization. It uses a general linear model to regress out any confounding effects from the features that are extracted by a layer. And when you did that, they then tried it on several data sets, or presented several data sets, one of them being one that consisted of 4 cohorts: controlled, HIV, mild cognitive impaired, and hand subject. That's HIV-associated neurocognitive disorder. And the data set was provided by UCSF, and the training data also got complemented by data from AIMI and SRI, which contain only controlled and mild cognitive impaired, and the SRI data control in HIV. And there were various methods that they compared against, just the vanilla 3D CNN, one with batch normalization, one with group normalization, and then the metadata normalization that we proposed. Interestingly, we achieved the overall highest accuracy with that approach, and the also lowest standard deviation in the accuracy scores across the 4 cohorts, and the correlation of the features with your confounder, in this case, the 3 studies, was the lowest. Also, you can clearly see from the embedding a significant difference between for example, here batch normalization and our proposed metadata normalization, where in the features when you project them into 2D, basically you have here nice clusters of essentially the different sites that then is not present anymore once we do this metadata normalization. So the take-home message from this from these last couple of slides is that you really need to be careful and account for confounders is really essential when you want to do inference with respect to neuroscientific data that is meaningful. The next couple of slides, I want to just shamelessly promote our lab and show you that we don't just focus on confounders, but that there are many more topics that we like to discuss. So as actually said initially, is the goal of our CNS lab is to identify biomedical phenotypes in order to improve the mechanistic understanding, diagnosis, and treatment of neuropsychiatric disorders. And to do so, we develop machine deep learning technology that's hopefully unbiased and is applied across biological, neuroimaging, and neuropsychological data. There are 3 components to our lab. The first one is to create these kind of analysis approaches, which we focused in this talk on confounders. Actually, another interest of ours is how to apply this deep learning technology to longitudinal data. And here, an interesting article that Qing Yu spearheaded and was just published in Medical Image Analysis is the notion of longitudinal self-supervised learning, where we are trying to learn what we call the brain age without having the chronological age of the subject, just by the sequence of images or MRIs that we acquired of a subject. And so the reason why we want to do this is here's an example where we have the chronological age, where we have subjects around the age 80, and you can see that their brains, are very different, especially here with respect to the ventricles that are normally associated with aging. The ventricles are much larger here than here. And so our estimated brain age for this 80-year-old is more like they're 62 years old, where this individual is much older. Now, on the other flip side, if we are ordering individuals by brain age and now look at the individuals with the same brain age, their chronological age is also very different. But again, those MRIs, essentially from a global overview, from a rough overview, look much more similar. And further confirming our findings is when we looked at differences between controls, those with mild cognitive impairment and Alzheimer's. Here is the brain aging development, essentially, of your control cohort in blue. This is of the Alzheimer group. And you see that they age much faster and more. If we are looking at the slope, so at the aging speed, then the mild cognitive impaired group, as anticipated, was between the control and the Alzheimer control group. Also, another project is how to do longitudinal consistent prediction, which Jia Hong just published at IEEE BMNI. She is a electrical engineering PhD student in electrical engineering, and is working also in radiology and with us here in the Department of Psychiatry. And a common problem, if you apply the kind of these deep learning model, is that the predictions that you get over time are very noisy. I call them kind of skyscrapers, where you get great variation. And what she did is nicely, she added a longitudinal consistently loss function to this model in order to model disease progression. And so here on the Encanda data set, we can now nicely see, we get now predictions that are smooth over time. Qingyu Zhao also worked on resting state, longitudinal resting state analysis, and how to get away from this kind of cross-sectional approach where we analyze essentially each, where we extract networks out of each visit individually or separately. And he essentially developed the model that where we could where the visits would all be analyzed together to get, again, more longitudinal consistent measurements from our data set. So, and he used this in order to show sex differences in functional connectivity, as well as the influence of alcohol on functional connectivity. The fourth project that I want to present is the one spearheaded by Asana Daily, who is trying to extract the movement automatically from videos in order, for example, to generate a gait score that's then used for diagnosis of Parkinson's disease. And he would like to combine this with resting state fMRI in order to refine diagnosis. The second component that we work on is developing software that eases collaboration across sites and where basically our collaborators can upload their data, and then we curate the data and distribute them, again, using open source software. This has been used by Encanda, was initiated by Encanda in 2013 and is in production since then. The fourth part is a study. We perform a variety of studies that are not just confined to the human brain, but we actually also look at red brains. We have looked at hearts and cover a variety of different applications and diseases. One of them is lately COVID. And so this is a finding that is currently reviewed. Just to kind of end the talk on this note, where in the Encanda cohort, we computed based on self-reported depressant symptom, we computed the CSD score. And what you can see here, since 2013, this depression score is slightly increasing, but is fairly stable. And this is to be expected as the cohort ages. But once COVID starts, we see a significant increase in the self-reported depression symptoms. Actually, the risk for clinical depression from before COVID to during COVID has tripled. And as you can see, it is a continuous effect that is continuously increasing, just not at that rate. So what we also found out that younger women were most impacted, and that prior COVID alcohol use and sleep behavior predicted increase in depression symptoms during the COVID pandemic. At the end, I would like to thank the many people that are involved in this work. I mentioned Jin Zhao and Asana Daily before. I also would like to thank Dolph Pfefferbaum and Edie Sullivan for giving me mentorship and guidance in this work, and the many students and research scientists, software engineers that work with me on this. So thank you so much, and thank you for your attention, and I'm open to your questions.